you're anything like me, you're probably hearing a lot about gene editing lately.
They have treated a newborn baby with a rare genetic disease.
Life of a baby with a rare genetic disorder has been saved.
A baby named KJ, born with a rare genetic disorder.
But what if I told you the technology that you and I are gonna see out in the wild first is not gonna be gene editing in humans for disease like cancers or genetic disorders, but in other living organisms that can also have their DNA edited? In this episode, I want to show you the 3 incredible technologies that have nothing to do with human gene editing, but are undoubtedly going to change the world around us, and you're gonna notice way before you see a gene editing drug in the pharmacy. But for that, we need to go outside. Let's go. I didn't come out here just to show you how beautiful Melbourne is in the autumn, but to show you where most of the world's DNA is found. It's all around us, but we don't really think about it as such. Take a look at this graph. It's the mass of all the animals in the entire world combined. It is nothing compared to how many plants, microbes, fungi, and viruses there are. We're gonna be talking about a few of these in this video, but let's start with this big giant, plants. In order to really understand what kind of genetic edits we're making in plants, I wanted to interview an expert. So I got in touch with Dr. Daniel Dunkelman. His research is all about synthetic biology and plants, and the things that he shared with me are not things that I expected here at all.
Some really good example is like if you have GLP-1 inhibitors, which are currently being used for control of diabetes, but more often than not, I guess weight loss. And so those therapeutics are protein-based, and they're proteins that are modified at 2 specific positions to make them more resilient in the cell and just improve their overall kind of pharmaceutical properties. Now, when you're producing proteins, there is a process that is called solid-phase peptide synthesis. Is a chemical process, and that process has 2 severe disadvantages. One of it is its scalability, so it's quite hard to scale, but people will succeed because there's a lot of money in that. But the much bigger problem is that the chemicals you're using, this process is extremely toxic and wasteful. The dream would be that in a 10-year perspective, we could kind of produce our tomato plant. We could equip this tomato plant with all of the molecular tools to build these modified proteins. So you can have your medication from a plant that grows. So more likely than you growing that tomato will be the tomatoes being grown by someone, and this is thoroughly tested, and then you buy your kind of additive that you can add to your food and to consume the essential medications. That's not going to be possible for everything, but I think there's going to be a couple of use cases.
Daniel went on to list all kinds of other things that labs around the world are working on in terms of synthetic genomes and plants. After all, plant synthetic biology can solve 2 of our largest problems as a society. One is food security and making more food for more people, which we're going to need, but also carbon capture, as plants are, after all, the original carbon engineers of nature. But one of the things that I really want to ask Daniel is, how is this different from GMO? And is this really different to what we've already known as genetically modified crops all along?
So this is a very good question, and it's one that I have 2 answers to. So I think we learned a lot. So when it comes to GMOs, one thing that us scientists have this, that what we need to do is we need to much more proactively communicate. So one of the things that are so essential to understand is that humans, ever since our existence and domestication, have been genetic engineer of plants. So can't stress this enough. We have engineered plants. We have done it through selection. The time scale that is extremely slow. I encourage anyone to Google this. Google how maize used to look like and go onto the field and see what maize is today, and that applies to almost all of our crops at this point in time, or all of the things that sustain us as a society. So these are genetically modified plants. These are genetically modified plants by humans. So that's super important to understand. In the first place, we will find ourselves in a position where we'll have to do some tough choices. And I think in terms of my personal perspective, going the GMO route is not a tough choice, because there is no conceivable danger as long as you think about what you're doing, and as long as you have regulation in place that controls that nothing is going completely haywire.
Genetically engineering plants has the capacity to change our climate and our food sources. But the next use case for genetic engineering is completely different. And it's something that you probably don't think about in a daily basis. But in the United States alone, over 100,000 people are on an organ donor waiting list. Over 17 people die each and every day waiting for one. And that could also be potentially solved with gene editing. Let me show you how. Scientists have been trying to use organs from animals, not humans, in order to solve that organ transplant gap for over 100 years. The very first xenotransplantation, the official name of transplanting an animal organ into a human, was in 1906, when a French surgeon attempted to transplant a kidney from a pig onto a woman's elbow, as she was suffering from kidney failure. But gene editing technology is giving hope to all these patients, because the biggest reason why we can't just take organs from animals and transplant them into humans, it's because our immune systems are not the same. Every single cell in your body has little tags on it. They're different types of proteins, but it essentially tells your immune system, hey, it's just me, don't attack me. I'm not a virus or a microbe or something you should be actively trying to hunt down and kill. Recently, surgeons and scientists at Massachusetts General Hospital had one of the most successful attempts at xenotransplantation because of this gene editing technology. It happened because of 3 reasons, and I want to show you each and every one because it's utterly fascinating. Pig kidneys are a great candidate for humans because for the most part, they're the same size as us and grow kidneys to the same size as us that filter the blood in a very similar way. But these proteins, those little tags on top of them, are still a problem. Scientists were able to make 69 different types of gene edits. So let me show you what they did. First, we need to take some things out of the pork kidney. Pork kidneys express certain types of sugar proteins on their surface that are not found in humans. So in order to do that, scientists needed to knock out those genes. And if you're wondering how did this happen, they don't just go and pick a pig from a farm just randomly. Those pigs were the result of IVF technology, where they create specific pig embryos, knocking out those specific genes, and then implanting them into a mama pig to grow. The second thing they needed to do is practically the opposite, and add specific genes that are expressed in humans, but not in pigs. These genes serve lots of different reasons. Some of them are for immune pathways, others are for coagulation factors that trigger a blood to thicken when needed, and others are involved in the inflammatory response. It shows you how complicated we are molecularly, and not the entirety of the animal kingdom is the same. But lastly, they need to do one more thing. They needed to edit out something known as PERVs, porcine endogenous retroviruses. This is a fun fact for all kinds of animals, but part of our DNA is actually viral DNA. These ancient viruses have embedded themselves into DNA, and they don't cause us any disease, although they're involved in some pathways, which is for another video. But pigs and humans have different types of endogenous retroviruses. So the final edits were super important because porcine endogenous retroviruses can infect human cells, whereas our human endogenous retroviruses can't. This is something that can only be achieved through gene editing technology. The 62-year-old patient that received this xenograft showed very promising results. Unfortunately, he passed away at day 52 for non-related reasons to the pork kidney transplant. And this is because new medical technologies need to be first tested on patients that have no other options left. This is usually a compassionate basis or studies that can only enroll people where other approved therapies are no longer an option. But nevertheless, they're critical into our understanding and allow scientists to keep improving. Genetic engineering and gene editing comes with a certain level of risk. And this is a risk that, as you've seen, scientists take very seriously, whether it's something we're planting out into the wild or manipulating in order to transplant it into a human being. There's a lot of checks and balances that need to be satisfied first. But this last technology that I will show you now is totally different. It's a type of genetic engineering that you can contain in a simple tank, just like this. But it's solving for a problem that we have had thousands of years. You see, this and this and this is environmental pollution from fabric dyeing factories. This is because we are no longer dyeing our clothes with berries and roots. This used to happen a long time ago. We have manuals from 2600 BC from ancient China showing how to dye fabrics. And this is something we've been iterating and tinkering with for thousands of years. But it all changed in 1856 when William Perkin, a teenager at the time, was trying to find a cure for malaria when he created the first synthetic dye. This was mauveine, a type of very bright purple. And it was based on coal tar. What William didn't know at the time is that he was starting a revolution, because fabric dyes have for millennia been a treasured item to trade between East and West. But all of a sudden, you could create it out of petrochemicals. Very soon after that, there's another revolution that is starting. Jacob Davis and Levi Strauss are joining forces to bring the beloved blue jeans to the entire world, an invention that is so popular even to this day. We are producing over 4.5 billion pairs of jeans every single year, and most of them will be dyed blue using a synthetic form of indigo. Indigo is a natural dye that gives off that beautiful blue hue. But what if I told you there was a way to use gene editing here too? And that is what Hue is doing. A biotech company that is growing a specific type of microbe in order to produce the perfect hue of blue. You see, nature already has the instructions on how to make these beautiful colors. Just take a look at the feathers of parrots or the petals of flowers. We can create a lot of natural, vibrant colors. So scientists were thinking, what if we take those same genes and insert them into the genome of a microbe? These microbes grow in a tank, just like this. And as they grow, feeding off a very simple type of sugar, they start producing the exact type of blue you want them to. This is revolutionary because to produce 1 kilogram of synthetic indigo today, you need 75 kilos of petroleum. And that is just the beginning. The fixing process also requires other toxic chemicals like formaldehyde and benzene. But with gene editing, creating this dye is more like brewing a beer. A lot more natural, less environmentally destructive, and doesn't take nearly as much water and land as traditional synthetic or natural dyes would. These companies are just the beginning of a revolution that's happening quietly. In the 1800s, we finally started to master industrial manufacturing and petrochemicals, figuring out a way of turning fossil fuels into plastics and dyes and furniture and all sorts of other things. But right now, we're going through a renaissance of synthetic biology, where we're figuring out ways of how to harness the power of natural organisms and all the tools we have for genetic engineering in order to create materials, pharmaceuticals, and all sorts of other things using the power of nature instead. I'm so excited to take you on this journey with me. If you liked this episode and love to learn about new technologies and the people behind them, make sure to follow along, because every week we will be publishing a new story on the incredible founders and the incredible startups that are building the technology of tomorrow.